So this chapter covers viruses, and there's a picture of the virus that causes the flu to start out the chapter. And in section 1 is titled The Nature of Viruses, and here as an outcome you should be able to describe the basic structure of viruses as well as their genetic makeup, their genomes, and then be able to explain why viruses are referred to as obligate intracellular parasites. And that just means that, number one, they harm cells, and in order to do that, they have to get inside the cell. So intracellular is within the cell. Now, all viruses are going to have the same basic structure. They're going to be made of a nucleic acid, which is where genetic information is stored, and then they're going to be surrounded by a protein overall. There is no cytoplasm, so these are not living cells. They are kind of on the fringes of life, but not. They don't fit the definition of what life is, but they are very important as they interact with living cells. Now, the nucleic acids can either be deoxyribonucleic acid or ribonucleic acid, and the nucleic acids can either be circular or linear. They can be single-stranded or double-stranded. Like DNA is double-stranded, RNA we learned was single-stranded. And RNA viruses can be segmented, and that just means that if it's an RNA virus and it's segmented, it's going to have more than one piece of RNA in there. I think, if I recall correctly, influenza virus has like 7 segments, 7 or 8, something like that. Now, viruses can be classified based on whether they're RNA for their genome or DNA viruses, or retroviruses, which we'll explain here in a bit what those are. So viruses, for the most part, are going to be basically a protein sheath that surrounds their nucleic acid, and the protein sheath is going to be called a capsid. So that's an important structure to know. And then on the inside, you'll have your nucleic acid, and the nucleic acid would be either RNA or DNA. As far as the capsid goes, the capsid is going to be made of at least one type or a few types of proteins, and these proteins are coded for in the viral genome. So the viral RNA or DNA has instructions for making those proteins. Now, the viruses may also carry special enzymes with them, along with the nucleic acid within the capsid there. An example would be the retroviruses, which carry an enzyme called reverse transcriptase. So reverse transcriptase is only found in retroviruses, and it gets its name because of what it does. You might recall from your Biology 1 course that DNA, our genes are found within DNA, segments of DNA that code to make a protein. And the DNA has to have its code transcribed to make an RNA or messenger RNA, and then that messenger RNA attaches with ribosomes, and then the ribosomes are going to make your proteins, right? And so the DNA going to RNA, that process is called transcription. And going from RNA to protein is translation, because it's at this point that we're going from a nucleic acid chemical language to a biomolecule made of different, it's basically different, it's made of amino acids. But the process called transcription is done by an enzyme called DNA polymerase generally. And viruses that are RNA viruses, they're called retroviruses because they go back. That discovery kind of changed, because before that we always thought that the only thing that could possibly happen in the flow of genetic information was DNA to RNA to a protein. And then when they discovered retroviruses, they have an enzyme called reverse transcriptase, and allows us to go from a viral genome that's made of RNA. So within the virus you'd have RNA, and the enzyme helps to go backwards and make DNA, right? So animal viruses structurally, so that's an example of an enzyme that can be carried within with the virus itself. And the instructions for making these enzymes are in the genome for the virus. So animal viruses, some animal viruses may have an envelope. around them. And so if we were to draw an enveloped virus, an example of an enveloped virus is the virus that's causing the current pandemic, the COVID-19 virus. And this envelope is made of a membrane that was taken from the host that produced it. So the viruses have to go into the host. The host cell makes copies, and when the viruses or the viral particles are coming out, they actually take some of that host membrane with it. And so it's the envelope comes from the host cell's membrane. So when it comes to viral hosts, the virus cannot exist unless you have a host that can make copies of it. And so again, they're obligated intracellular parasites. And about any organism that's ever been studied has been found to have viruses. This includes bacteria, humans, trees, anything living can have a host. So when it comes to the virus, though, they typically have a host range. So if you have a virus that infects plants, it's not going to be infecting animals, for example. And the host range is typically limited in those cases. So you might have viruses that affect only a certain group of animals, say certain types of mammals, for example. And even then, when the viruses are infecting multicellular organisms like an animal or a plant, they have what's called tissue tropism, and that means they can only infect certain types of tissues. And what that means is those tissues have cells that on the surface they're going to have these receptor proteins that typically receive the normal kinds of information, but what happens is the virus evolves to interact with those receptors, and when they do, they trick that host cell into taking in the virus. And so there's this tissue tropism. Examples of rabies has a tissue tropism for nervous tissue. The influenza virus, the right kind of cells are found in the respiratory tract, deep within the respiratory tract. So when you take those viral particles in, the receptors are in those cells within there. The cold virus, the receptors tend to be higher up in the respiratory tract. When an infection does occur, some viruses may lay dormant within the host cells, or latent is another way to refer to that. And an example is chickenpox. It was a common childhood disease. When I was a kid, I got it. And now they have a vaccine for it. But if you were ever infected with chickenpox, you never really get rid of it. The viral genome is a DNA genome, and it goes in and becomes part of cells in your nervous system. And then under certain conditions, that virus may reemerge and cause disease again. And in some cases, the disease is the same. In other cases, the disease is different. So in the case of chickenpox, that same virus will cause a condition later on in those individuals called shingles. And they're very uncomfortable. They create shingles creates very uncomfortable lesions there at the surface on your skin. And so, again, every kind of organism is affected by viruses, and usually more than one. So there are a lot more viruses than there are organisms. Now, for viral replication, the virus itself, it's not alive, but the virus has instructions for the host to make copies of it. And so the virus basically goes in, hijacks the metabolic machinery, the enzymes from that host, and gets the host to make copies for it. So the host is tricked into making copies. And oftentimes when those copies are being made, the host cells are damaged. So if you're a single-celled organism like a bacteria, and you have a virus that attacks the bacterial cells, when the viral particles emerge, they damage and kill the cell. In other cases, the cell may not die, but the production of the viruses does create diseases in the case of multicellular organisms like human. for example, when you get the flu. Generally speaking, when the viruses emerge out of the cell and break out of the cell, there's no active metabolism in there. So that's what this statement means. They're metabolically inert. And we're going to refer to them as virions. So if you ever see the word virion, it's a virus it hasn't gotten into a host yet. It's a viral particle. Viruses, again, they don't have the tools for making copies of themselves, so they can't reproduce on their own. That's one of the reasons why we can't say viruses are living. They're kind of on the fringes between non-living and living. This way I like to refer to them. They don't have their own ribosomes, so they can't make their own proteins. They don't have enzymes, the enzymes necessary for making those proteins, as well as being able to synthesize nucleic acids as well. In some cases, they are carrying enzymes to help with that process, and oftentimes that's an important part of the viral cycle. Now, the virus, when it goes in there, it's going to basically take over the metabolism of the host cell. If the virus had any intentions, the intention is to get in there and take over the machinery that does transcription and translation. So remember, transcription is from DNA to make your RNA, and then from RNA to the ribosomes to do translation. And so the viruses will typically have different sets of genes. There'll be early genes when they first infect the cell, middle genes, and all these genes are geared towards making certain proteins that are important in the process of making the parts to put more viruses together. The late genes are going to be important in assembly of the capsid overall, and also proteins to help that virus get out of the host cell once they're ready to emerge from there. And so the end result of the expression of all these viral genes within their genome is the release of new virions to go infect other host cells. Viruses come in different shapes. There are some basic shapes that the capsids will produce. For example, the tobacco mosaic virus, which is TMV, the abbreviation for it, their capsid is a helical structure. You can see that over here. You can see the individual components, the individual proteins that the virus genome programs to make. When those proteins are made, they in most cases, they're going to assemble spontaneously, and they'll assemble in very specific kinds of shapes. And this one is called a helical shape. There you can see that the tobacco mosaic virus is an RNA type virus. And then you have the icosahedral shape, and it's somewhat spherical, and this kind of maximizes the amount of space inside for your viral genome. In this case, we have a DNA virus called the adenovirus, which is an animal virus. But the icosahedron, the protein subunits assemble into these units that are triangular in shape, and so you get 20 of these triangular units, and it gives you this icosahedron geometric shape. The viruses may be naked, which means they're not going to have an envelope that they get from the host cell. An example is the polio virus would be naked. Some viruses are going to have very complex capsids. The pox virus, for example, is a multi-layered capsid. And then you have the viruses that we generally call bacteriophages, which translates to bacteria eaters, because they infect bacteria, and then when they emerge, they kill them. And so these T-even phages have a binal symmetry. And you can see that over here. What's meant by binal is they have two major types of components to it. They're going to have the icosahedron, and then they'll have this helical tail to them. And in this case, you can see that DNA genome inside that bacteriophage. And then a virus like influenza is going to have an envelope, which is pictured here on the right. So this is your influenza virus. It has a helical capsid in there. Influenza is an RNA virus. And here you can see the envelope that the virus, when it was produced, when it emerged from the host cell, ended up taking some of the membrane with the cell there. Here's just a picture of one of those bacteriophages. On the left is an electron micrograph, color enhanced. They're usually black and white. Don't labor too much on memorizing all the different parts of what a phage virus would be, but you should know essentially the basic symmetry is binal. It's got that icosahedron and the helical tail. Some of the other structures are important for attachment to the bacteria host and so on, but binal symmetry is definitely one. This image just shows the relative sizes of your viruses compared to some of the cells they might infect. Here you have a eukaryotic cell called a yeast cell, and here's a bacterium called Streptococcus, and here is E. coli, which is a rod-shaped bacteria. And when it comes to eukaryote and prokaryote, we're going to use a scale on the order of micro, or microns, or micrometers. And micro is 10 to the negative 6. 10 to the negative 6 means 1 over a million, or 1 millionth of a meter. So if you took a meter stick, you broke it up a million times, that's a micrometer. So yeast cell has a diameter of about 7 micrometers, Streptococcus 1 micrometer, and the E. coli about 2 micrometers in length. But when you look at viruses, micro is too big of a unit, so we're going to go to nanometers. And nano is not 1 over a million, but 1 over a billion, and that's 10 to the negative 9, so that's really small meters here. So you can see viruses like the herpes virus is 150 nanometers, and then some of the other viruses in size. And they're going to compare it also to a protein called hemoglobin, which is found in your red blood cells. That has a diameter of about 15 nanometers. And here's a T2 phage virus that infects bacteria. So you can see them landing on the bacteria and their relative sizes relative to each other. Now, viruses do vary in the kinds of genomes I mentioned earlier. There's RNA viruses, there's DNA viruses. They may be a double-stranded or single-stranded. You can even have double-stranded RNA viruses. So when RNA viruses are single-stranded, we might abbreviate them like this, SS for single-stranded, RNA. And the thing about RNA viruses include influenza, by the way, measles, and even the common cold. And as they go into the cell, they're replicating. They also have to replicate their genome because they have to be able to put that into the new viruses that are being made. And it turns out that the enzymes that they're using to do this are not very faithful in making copies of themselves. So the enzymes that do this are very error prone, and that results in high rates of mutation. And that ends up making RNA type viruses difficult targets for your immune system. If you get sick with an RNA type virus and then your immune system clears it out because your immune system made antibodies, well, your immune system recognizes that version of the virus. When the virus mutates, maybe in another host, maybe in you, it mutates, and then you get exposed to that mutated form. Your immune system didn't recognize it because it went through some changes. The outside has literally changed in a way that your immune system didn't recognize it. So that makes it hard for your immune system to recognize it. It makes it hard for vaccines and drugs to treat these types of viruses. And then some RNA viruses are referred to as retroviruses. These retroviruses are single-stranded RNA type. And what happens is that the RNA genome is going to be reverse transcribed to make a strand of DNA. And so these viruses are carrying their own enzyme called reverse transcriptase, which was mentioned earlier. Remember, it's DNA to RNA. And RNA, your messenger RNA goes to the ribosomes. Ribosomes bind with the RNA to make your protein. But right here, your viral RNA is reverse transcribed to make your DNA. And an example of a retrovirus. These are called retroviruses. An example is HIV, which stands for human immunodeficiency virus. Now, most DNA viruses are going to be double-stranded, and those are going to be the viral genome is replicated within the nucleus of the eukaryotic host cell, and they include smallpox and herpes simplex viruses. Here's a table showing some of the viruses that affect humans and cause disease. The name of the disease is here on the first column, your pathogen, the type of genome they have, and then the vector and epidemiology. The vector is how it's transmitted, and then the epidemiology is basically how many people in a population can get infected with it, and when they get infected, how many get really sick, and then in many cases, some will die from that viral infection. So epidemiology studies those types of things. And indeed, we're hearing a lot about that with the COVID-19 virus. So your chickenpox virus is a double-stranded DNA virus. And again, there's a vaccine's been available since 1995. So if you got that vaccine, you never got chickenpox the way I did. Herpes, the disease called herpes is caused by HSV or herpes simplex virus. That's also a double-stranded DNA virus. And this one is easily transmitted by skin contact when viruses are actively being produced and released through blisters. Then you have your smallpox here. It's also double-stranded. This one was very deadly. Killer historically. It was eradicated in about 1977 when vaccines were widespread and given to populations worldwide. Going to your AIDS disease, that's caused by human immunodeficiency virus or HIV. And this one is not a DNA double-stranded, it's a positive and that's something that requires a little bit of explanation. It's a positive single-stranded RNA virus. And all that means is that while we still have to make copies, when that genome gets into the host, we got to make copies of this. The genome itself can be directly translated. In other words, this can serve as an mRNA, the purpose of an mRNA. And the mRNA basically binds with the ribosomes to directly go and make your proteins. There is another kind of single-stranded RNA category that's negative single-strand RNA. And here, this one, it cannot serve as an mRNA. Instead, an enzyme is needed to take the sequence of bases on this RNA strand and make the complementary strand that then serves as your messenger RNA to make the viral proteins. So HIV is a positive single-stranded RNA virus. So is polio. Again, I'm going to suggest that you read over this once. Don't go memorize the things. You'd be wasting your time doing that. But go over and read through, scan through, and read the epidemiology of these. So moving on, just going to skip to Ebola. You've probably seen or heard about Ebola in movies and so on. And this one is a negative single-stranded RNA, which I already mentioned earlier. The RNA within the virus can't be directly made into a protein. And here, the thing about Ebola is it sometimes is the disease manifests itself, Ebola, as an acute hemorrhagic fever. And hemorrhagic means you're bleeding, and you bleed like everywhere. You're bleeding in all your connective tissues. You're bleeding from your eyes, from everywhere. And there's a very, very high mortality rate, from 50 to 90%, depending on where the outbreaks were. Your influenza, it is a segmented, there it says 8 segments, I think I said 7 or 8 earlier. And it's a negative single-stranded RNA, like we mentioned before. And all the rest on this list are all negative single-stranded, including rabies, which has the tissue tropism for the central nervous system. And here, SARS, the disease, severe acute respiratory syndrome, was caused by a coronavirus type virus, just like the COVID pandemic that we are having right now. I think the current coronavirus that we're seeing is transmitted a lot more easily, and apparent at this point, the data suggests has a much higher fatality rate when you do have an infection. So that's the reason why we're applying the precautionary principle overall. Another interesting thing is that we've seen these giant viruses that have been recently discovered, and they kind of blur the lines between the non-living viruses and living cells, because viruses just aren't alive. They have to take over the host, right? Most viruses are going to be pretty small, less than 250 nanometers in size. A virus called they named Mimivirus was mistaken for a bacterium. And that one had a diameter of 750 nanometers. So that's 3 times bigger than some of the largest viruses that we were seeing. And not only that, its genome is relatively large with 1.2 million nucleotides long. When you say MB, it stands for megabase. Remember, every nucleotide within your nucleic acid has a base, whether it's adenine or guanine or cytosine on there. So, and even larger viruses have been found more recently. They've called them Pandora virus, Pithovirus, and Closnavirus. And the thing about these is where they really start to blur is not just size, is that they're carrying even more metabolic machinery, even some of the molecules required for translation. So within their genome, they have the program for making some of the tRNAs, the transfer RNAs that are important in translation. So that's pretty interesting discovery, which they're starting to study. So this section touches a bit on viral diversity and different types of viruses out there. So your learning outcomes here are to compare different classification systems for viruses and to describe the role played by area of study called metagenomics in describing diversity. So when it comes to trying to get a handle on the different types of viruses out there, it's helpful to classify them. And so there's different ways that viruses can be classified. And indeed, there's a formal classification system or a taxonomy that's organized by the International Committee on Taxonomy of Viruses. And there they use similar taxonomic ranks that we see in classification of other organisms. They use order, family, subfamily, and genus, and they'll even identify species, although there's problems when it comes to trying to identify a virus by species. They don't use higher ranks, so you won't see class or phylum or kingdom in their system. The interesting thing is that some genes within viruses are conserved. They must be pretty important for the virus to be able to replicate within a host. And if that's the case, that lends the viral genomes to phylogenetic analysis, which was a topic of earlier chapter for us here. And so analyzing similarities in their genomes can help guide that classification. And so I was just curious, because we're in the middle of a pandemic with COVID-19, which is a coronavirus, which is actually, as I mentioned earlier, is related to the disease SARS. It's caused by a coronavirus type. There's another one, CoV-2 for coronavirus 2. And they were studying genomes there from viruses collected worldwide. And so this is an actual journal article published recently in April of 2020. And I just thought it'd be interesting to put that there. Another way we can classify viruses is by the diseases they cause or the hosts they infect. However, these two ways of doing that do present problems, which I'll mention in the next section or the next slide. And then the one that seems to work out a bit better is based on the way the genes are expressed, whether they're a DNA virus, RNA virus mentioned in the earlier section. And this scheme for classifying is called the Baltimore classification system. So looking at classification by disease or host. Now, both of these methods have significant limitations. First of all, if we're going to talk about trying to classify by disease, not all viruses cause disease. So that can be an issue. How can you classify it? If a virus can infect a host, and then doesn't cause any problems for the host. Some viruses also cause different diseases under different conditions. The virus that causes chickenpox, for example, when you get it once, then later on it goes dormant or latent in your nervous tissue and then comes back later and then causes another disease called shingles. So that could be a problem. Same virus, different disease. And then there are other viruses that give the same effects, or the same symptoms, the same kind of disease. For example, when you get the cold. The common cold is caused by several different viruses. So that, how can you classify a virus as a cold virus if more than one virus can cause cold symptoms? And then some viruses, if it comes to classifying by host, some viruses infect different types of organisms. And the example is influenza. Influenza affects birds, humans, pigs, and so on. And so that makes the host classification by host challenging. So the one that seems to have more of a broad ability to classify different viruses is by the genome expression. And that one is the Baltimore system. And so the Baltimore system is based on the structure of their genome. Is it DNA or is it RNA, and how those genes are expressed. And so there are different groups identified here. We've already talked about some of them. The single-stranded RNA viruses here, which I mentioned in the earlier section. You have your plus single-stranded RNA viruses, and then your minus single-stranded RNA viruses. And then right here you have your retroviruses in the third one here. You got your retrovirus. And then this group here, group 7, is a more recent description or group. And that one's interesting because it is a DNA type virus. It's a double-stranded. It has both the sense and the non-sense strand, both strands as well. But the thing about this one is the DNA is not, it's in segments or in pieces. So it's not one full double-stranded DNA. And that is something that makes this new discovery group unique, and for the time being, it's not a very common class of viruses. So coming back over here, you have your double-stranded DNA type viruses. Those would be group 1, and you have both strands. Now, one of the strands, the positive strand, would be the one you're going to have to transcribe it, right? So this arrow here would represent transcription to make your mRNA. And because this is the viral DNA, then the mRNA has the transcript. It has the codes or the sequences that are going to build the viral proteins. And then you have, and this is different than what we talked about in Biology 1. We always talked about DNA being a double helix, right? But some viruses don't follow that, and they're a single strand of DNA and within the capsid. And so they're going to have enzymes that would put together your other strand of DNA, and then one of the strands there is going to be used to be transcribed using an RNA polymerase to make your messenger RNA, which now has the codes for making your viral proteins. And then something also different from your Biology 1 course, you have double-stranded RNA type, so that's something we hadn't seen before. And then you have your plus and your minus, and the plus strand can be used directly to make your messenger RNA. And then you have your plus and your minus single-stranded DNAs there, RNA type virus, sorry. And there, if you're a plus strand, then your strand can actually directly go in and serve directly as your mRNA to make your viral proteins. However, keep in mind, all of these viruses do need to also make copies of their genome, so they need to have copies of their DNA or their RNA genome to be packed inside of the new viruses that are being produced before they're released. And so that's going to require special enzymes, especially if you're an RNA virus. You're going to need an RNA type of polymerase that doesn't use DNA as a template, but instead uses RNA as a template. And that RNA polymerase would be used to build more of these viral RNA strands for their genome. So when you're a single-stranded RNA you're going to have to build the other strand, the complementary strand. So the replication is similar to DNA replication in that you use one strand as a template to make the other one. And that's why they have the two arrows here for the group 4. One is to directly, the RNA can directly serve as mRNA, but you're going to make lots of negative strands. So the negative strands can be used to make lots more of the RNA. Not only just to be packaged within the new viruses, but also to go in and make lots of viral protein. So you've got to make lots of copies so that you can make enough protein to make a lot of viruses. So the same is true for all of these other viruses. You're going to need those enzymes. In some cases, the enzymes are brought into the cell by the viral genome itself. The viral genome codes for those enzymes, special enzymes. Now, keep in mind, remember that the RNA-type viruses, the enzyme, the polymerase is used to build copies of those are error-prone, so these RNA viruses tend to mutate. Remember, the retrovirus itself is going to be a positive strand RNA. It's going to use a special enzyme called reverse transcriptase to build your DNA molecule, and then becomes double-stranded. And then one of the strands would serve to make your messenger RNA that would be used to produce your viral proteins. Now, there's a new area of study called metagenomics. It's advancing a lot more now. It's not brand new, but here it's always been the case where you cannot isolate sometimes and grow in a lab and culture in a lab. Certain bacteria just won't grow. And the same for viruses. Viruses they're not always easy to culture in the laboratory. For example, if you're going to try to culture viruses that infect bacteria, a lot of times it's not. They haven't been able to do it for many. And how do they know this? Is because they go and they sample the environment. They'll sample soil or water or air, anywhere you can find DNA. And they take that DNA and then they use polymerase chain reaction technology to make multiple copies of that strand of DNA, then analyze it, and then they compare it to known genomes from viruses or bacteria, for example. The focus here is on viruses in this chapter, but same thing. And what they find is that a lot of that DNA doesn't match anything that's been described, and that tells us that these are bacteria or viruses, like for this chapter, that they haven't been able to culture in the laboratory, and that they exist somehow in the environment, and they're interacting with other living things. We just haven't been able to culture them. So this has opened up a whole new area of study that has gained a lot of interest in biology. So in this section, we're going to be looking at bacteriophages or bacteriophages, and these are going to be viruses of bacteria. So they infect bacteria. Your learning outcomes are going to be able to distinguish, and you should be able to describe actually both lytic and lysogenic cycles when these phages are going through their reproduction cycles. And then we're going to see how viruses can contribute DNA to their hosts. Again, bacteriophages are going to be those that infect bacteria, and they are diverse. It's a diverse group of viruses. They're going to be called phage for short, and the thing that they'll have in common is that they infect bacteria. One that's been well studied is E. coli, which is short for Escherichia coli. This is a bacteria commonly found in your large intestine, which is where coli comes from. The specific epithet is reference to the colon. Well studied are the T series phages, T1, T2, and so on. And they're the ones that have that binal symmetry. They look like they have the head and the tail. The head is icosahedron and the tail is helical shape. They've also found and discovered that the archaea also have their viruses. Remember the archaea, there's two domains for the prokaryotes. There's the bacteria and the archaea. The archaea many times are found in extreme environments. So viruses have been discovered for the archaea, and they're still in the characterization stage for this group of viruses for the archaea. So looking at the reproductive cycle, I thought we'd just go straight to the diagram and then see what the slides with the text say about it. So there's two ways in which the bacteria, the viruses can reproduce themselves. And there's two cycles. The first one is the lytic cycle, and the second one is the lysogenic. So lytic cycle and lysogenic. And in one case, the viruses are replicated, which is the lytic cycle. And when the virions exit the cell, the host cell, they basically kill the cell in the process. In the lysogenic cycle, the bacteria is not killed right away. Instead, the viral genome goes into a latency where it just lays dormant inside the bacteria host. So let's take a look at the different stages. The first stage is attachment. The phage has to attach to the cell wall where special proteins helps attach. And then some special proteins are going to allow for the second stage, which is penetration. Now, these two steps are required whether it's lytic or lysogenic. You have to get your bacteria DNA into the host bacteria. So let's say that this virus is going to go straight to making copies. So the viral genome gets placed within the cell, and the viral genome begins to be transcribed and translated, and we get into the synthesis stage. And here you're going to start making more copies of your viral genome. You're going to be making the proteins that are necessary for putting together a virus, including the proteins for the capsid and any other special proteins that may help the virus get into a cell or get out of a cell here. So you can see the next stage would be once we've made a lot of the proteins and copies of the genome, in this case the DNA, viral DNA. Now we're going to assemble them, and so you're assembling your parts, and eventually you get your functioning viruses that then are released from the cells. So the last stage there would be release, and these virions now are free in the environment to go and infect other bacteria cells. Again, and at this point here, the lysine of the cell kills the cell in the process. Another part is a possibility is when the DNA penetrates in there, instead of going into the lytic cycle, the virus enters the lysogenic cycle. In this case, a special enzyme is going to be produced that gets the DNA and integrates the DNA into the circular chromosome of the bacteria. So that would be integration. Okay? And then after integration, then propagation would occur. And propagation is to reproduce. So here now, the bacteria is going through its normal reproduction. It's going through binary fission. And before you divide, a bacteria divides into two cells. It has to make a copy of its chromosome. And what it's actually doing is making a copy of the viral chromosome as well, or the viral genome. So every time the cell divides, we're making more copies, and we can keep going on and on as those cells divide. In that bacteria population, they're actually multiplying the virus, but the virus is dormant at this time. It's latent. Now, the virus at this point would be called a prophage. So that red segment of DNA right there, that's your prophage. More generally, we would call it a provirus. It's not a virion, it's not a virus, it's in its latent period. And as far as the bacteria that are carrying the prophage, those are referred to as lysogens. Okay, so the bacteria itself is called the lysogen, and here they call it lysogenic bacteria. Same, lysogenic is just the adjective form of lysogen. So here you have your lysogens are going through just binary fission, propagating themselves, growing as far as the bacteria population goes. At some point, though, some of these lysogens, these bacteria that are carrying the prophage, are going to go through induction. And induction can be caused by some stress in the environment, maybe low food availability, perhaps radiation or something, and that's going to trigger the virus, the prophage, to go through induction. And now we're going to start transcribing and translating genes that are going to start synthesizing the parts to make your bacteria. Now you've entered the lytic cycle, and then you go through, and the lytic cycle happens as it normally would. So those are the two ways in which your bacteriophages can replicate. Now, if we go to the next slides, all they're doing is just giving you those same stages for the lytic cycle. We start with attachment and then penetration, which, by the way, you need to start any one of the cycles. Lytic continues with synthesis. These were all mentioned in the prior diagram. Then we have to assemble and then release. Now, something not mentioned that was on the diagram is the eclipse period. So during the eclipse period, this is going to be the time between absorption when the virus has injected its viral genome into the bacteria, and then the time from that time on to the time in which the new particles are being produced. That's called the eclipse period. They're not being released yet. If at any point during this time the bacteria cell lyses, which it tears apart for some reason, it breaks apart, and the viral particles have not fully assembled, then you're going to have very little virions, because, and which means they're not going to be active and be able to infect other viruses or other bacteria. Again, when you go into the lysogenic cycle, which was the other part of the cycle there that we don't kill, you're not going to immediately kill the cell. That's called the latent phase, and the virus, the prophage is integrated into the host chromosome, right? And so they call it the lysogen or lysogenic phase. So another term here is lysogen. Okay? So those are all illustrated there on that diagram. There's some good animations as well that I have linked on the activities page that you looked at to click onto this video here. There's a very well-studied phage called phage lambda, and also infects E. coli. And this one has been well studied for the lysogenic phase part of the cycle. And in this one here, they've discovered, well, how does the virus how does it determine whether the virus goes into lytic or lysogenic? And it turns out when penetration first occurs and the viral genome enters into the bacteria, some of the genes are already going to start being transcribed, and some of the early genes that are transcribed are these regulatory viral proteins. There's two kinds. One of them is a regulatory protein that gets and initiates transcription of the viral genes that are going to get the lytic cycle to start. So you start cranking out virions and killing the bacteria cells. The other regulatory protein is one that's going to be involved in stopping the lytic cycle, and also even producing an enzyme called integrase. And integrase is an enzyme that allows the viral DNA to be integrated into the bacterial chromosome, and then you get into the lysogenic cycle. So these two regulatory proteins are going to be produced, and depending on the conditions, you're going to have one that turns out to be the winner. So if it's the regulatory proteins for the lytic cycle, then you get into the lytic cycle. If it's the other one that's going to produce integrase, depending on those conditions, then you end up entering into the lysogenic cycle. In any case, if you get into the lysogenic cycle, at some point, some stresses are going to induce. It's called induction, which we saw in that diagram earlier. So come back to the diagram here. Yeah, at some point in the lysogenic stage, we're going to go through induction, okay, and then get into the lytic, go into the lytic cycle. So when it comes to the lysogenic type of cycle, phenomenon has been observed called phage conversion here. And in this case here, the bacteria's phenotype is actually changed by the virus. When the viral genome gets integrated into the bacteria's chromosome, some of the genes from the viral genome start to get transcribed and translated. And in some cases, the products of the viral genome are toxic to some other organism, right? So they give an example with a bacteria called Vibrio cholerae And this one causes a disease called cholera, which infects the lining of your intestine and causes severe diarrhea, and death can happen from this. This is usually in places where there's not good sanitation and water's contaminated, and people who live in those areas don't have a choice, and they drink the water and they get sick from this. But it's been discovered that there's a virus that's called CTX, and I think the Greek letter is Rho. That's the name of the virus. It has a gene in there that produces the CTX. The CTX gene produces this toxin, which changes the phenotype, making the bacteria pathogenic when normally it's normally not. So this is what phage conversion is. It just changes the phenotype from a harmless bacteria to one that's can cause disease. So in this section, we're going to look at viral diseases that affect humans, and your learning outcomes here are going to be to describe the difference between acute and persistent infections, and then relate antigenic drift and antigenic shift to seasonal flu outbreaks and pandemics. This will help us understand how you need a flu vaccine every year. And then look more closely at chronic and latent viral infections that we see in humans. Now, when it comes to human viruses, they can be categorized by how rapidly and frequently the virus is produced, and on the appearance of any symptoms associated with disease. So there are acute infections. Let's skip to that one first. So the acute infections, these would be rapid, and the replication of the virus replicates rapidly, and you get a sudden onset of symptoms. And these are exactly the kind that lead to outbreaks, epidemics, and pandemics. And that's seen here on the top graph here. Here you can see if we look at time, the virus begins replicating, and then you have a period where the onset of disease. And then if your immune system ends up taking care of the virus, and then the virus has stopped being produced and the disease goes away. Examples of acute include influenza, rhinovirus, which are cold viruses. And then you have your chronic and persistent infections. For chronic, they would be like HIV or hepatitis infections. You get infection and the virus increases. The number of virus increase. You may not get a disease, and so over time you have this chronic infection, and then all of a sudden the virus is increased further, and then disease sets in. And so again, this is the case with HIV and hepatitis type infections. And then you have latent infections, and an example there is the herpes simplex virus, or the HSV viruses. Here, your initial infection, you increase, you get the disease, and then the virus will hide in some tissues and become latent, and no new viruses would be produced. So it would be become part of a chromosome and be called a provirus at that time. And then sometime down the road, the virus comes out of latency and begins to produce more virus, and then you get disease again, and then goes back into latency, and then disease again, and that's exactly what happens with things like cold sores and genital herpes. And then there's one more here on the bottom that'll be covered in the last section. When you have infectious agents like prions and other cancer-causing viruses, over time you're going to have a buildup of those agents, and then eventually progressing to the disease like a cancer or wasting away of your nervous system in the case of prions. We'll look at prions in the next section. So the first human virus we're going to look at is influenza, and this is one of the most lethal viruses in human history. It was a pandemic in 1918 and 1919, and it's thought to have infected about a third of the world's population, and that resulted in tens of millions of people dying. The flu virus is an envelope virus. There's three main types. There's type A, type B, and type C. Type B and type C are just human type viruses, and these tend to give more mild symptoms. Sometimes you don't even know that you maybe have gotten the flu from those. And then you have type A, which are more serious. These are the ones that lead to epidemics. And these are the kind that can infect humans and other animals. And that's the problem there is in this case, you get new combinations of flu because the same animal can be infected with two different strains of the virus, and then it mixes up things. We'll look at that in a little bit. Now, the flu virus, they vary. They have different types based on two proteins found on the envelope itself. There's the H protein, which stands for hemagglutinin, and then uramidase protein that's found on the surface. And both of these proteins are coded for by the viral genome. And the H protein spike helps aid in the entry of the influenza virus into the cells that are found in the respiratory tract. And then the uramidase aids in the exit. So both are very much needed to complete the cycle of the influenza virus. So there's two things that are at play here when it comes to your flu virus, and if we look at the type A virus here, and one of them is antigenic shift. Now, antigenic shift is referring to those proteins that are found on the viral envelope, those protein spikes. They're also referred to as antigens because those proteins are antibody generating things. They don't belong in your body, and your immune system recognizes them not being there, and you produce proteins called antibodies that will go and stick to these viruses and basically immobilize them and tag them for destruction. And the problem here is that the flu virus, it's a segmented virus and it's an RNA virus. So remember from earlier section, RNA viruses, when their genome is being replicated, as the virus is being replicated within the host cell, there's a tendency for the enzymes, the polymerases, for making copies of the RNA to not do it so well. So you end up with mutations. And so that leads to an antigenic drift. And the surface proteins go through little changes because of these mutations. And the end result here is, this is supposed to be a model of an antibody that your body's produced, and they stick to the normal or to the either the H or the N protein spikes from the infection initially, and then helps get rid of the virus infection that you have. But during many replications, a mutation may have occurred in that the spikes are now different. The protein spikes have modified now, and the antibodies you're carrying from last year's flu infection are not going to stick because of that change. And so this is one reason you need a new vaccine every year because of those random mutations that take place, right? So this is why those vaccines, we've got to try to develop a vaccine that's for the next year's most common type of flu that's coming around now. There's many different kinds of H and N, and so they'll refer to them as H1N1 or H3N5 and so on, depending on what kind of protein spikes they have there. And then the next one is the antigenic shift, which has resulted in pandemics, and that's the bottom diagram here. So looking down here, in this case, you have a flu that can, a type A flu, that can affect birds and humans and so on. And the problem here happens when you have different strains that infect the same host. And so here you have an avian influenza with its H and N spikes for it, and that infects something like a pig, and then the human, and both come in contact with this pig, and so the pig becomes infected, or the animal becomes infected with both flu viruses. And remember that this is a segmented virus, so there's 8 pieces of RNA in there, and 8 pieces of RNA there. And so when these viruses are being replicated within the cells of the pig, it's possible that the new viruses that are being produced within the capsid end up getting some variable combination of the two types of RNA segments that are there. And that leads to having a shift in the combination of antigens that you have on the surface of your. So this is independent of just having the mutations occur here. No mutations have to occur. You just end up getting a different combination of the H and the N antigens on the surface here. So you can see here you have red and orange spikes, just to kind of highlight for the bird there, or the duck, and then for the human they have these blue and purple spikes. And then within the pig some new combination occurs, and you end up with a virus that has the red spikes from the bird influenza and the purple spikes for the human influenza. And so now you have this new flu virus that has a great potential to make an individual very sick if you get the infection. And so this is what will lead to those major epidemics and pandemics, because you have some new combination that our immune systems just haven't seen before. So this has definitely been the case for your pandemics of influenza. In 1918, they called it the Spanish flu, and it's a type A, and that's the antigens there in that combination are H1N1. Like I said, it killed tens of millions of people worldwide. And then there's a 1957 type A flu with some new combination with H2N2. And then there was a H3N3 in 1968, infecting 50 million in the U.S. and killing 70,000. So these are flus that this new combination, because of antigenic shift, can create a new combination that can be more deadly and spread as it spreads through the population. So now we're going to look at HIV, or human immunodeficiency virus, and here HIV leads to the disease called AIDS, or acquired immunodeficiency syndrome. And so the disease is different than the name of the virus. The virus is HIV, which stands again for human immunodeficiency virus. And this is a chronic infection that we saw on the graph earlier. And here, over time, the HIV is going to infect cells that are found for the immune system. These are cells that carry a receptor on the surface, a protein receptor called the CD4 receptor. So those are called CD4 plus cells. And they include macrophages. So these are large white cells that go around gobbling up bad guys like bacteria and other things. And then the T helper cells. These are going to be really important in communicating an immune response that ends up leading towards the development or production of antibodies for very specific pathogens that invade your body. So once you get exposed to them, the T helpers help make those antibodies from your prior infection. Now, what happens here is that if the HIV replicates and infects more and more of these cells, eventually the major cell that it really has a big impact on is the T helper cells. And for whatever reason, some of the other immune cells don't get killed by the replication of the HIV virus, but the T helper cells do, and those are the important ones. Those are the ones that are important in building up your immunity to a disease. So ultimately, the individual doesn't die from the HIV itself, but from some other infection that normally wouldn't cause death or disease. And these would be referred to as opportunistic infections. So the individual may die of cancers or some other kind of infection that your immune system might normally help fight off. So looking at the HIV cycle, here's the words on the left are describing what we'll see in the diagram here. The first thing, of course, is attachment. You're going to have receptors that are located on the surface of your cell. This is a CD4 cell. And one of the receptors, the purple one, you can see labeled down here, is the CD4 receptor. And the thing about HIV is it requires a co-receptor. And there's two types of co-receptors. They're the CCR5 or the CXCR4. These are just proteins, and they are given the name based on the name of the gene. They name the genes with these codings on there. And so you have to have those protein spikes, very specific protein spikes called GP120 spikes that are on the envelope. These are proteins that the HIV genome codes for. And so with both of those receptors, that's going to allow the virus now to gain entry. So the virus, once both receptors are attached to that GP120 protein that's on the envelope of the HIV virus, we end up with endocytosis. So now the capsid gains entry, and it's an RNA virus. This is a retrovirus. So the RNA gains entry into the cell, and then in the cell, you got to remember that these retroviruses are carrying that reverse transcriptase with them, so they release the reverse transcriptase with them as well. And the reverse transcriptase is going to go and take the RNA, and in step 3 here, we're going to start to make DNA from there. So we're going to reverse transcribe the RNA to DNA. And first, the reverse transcriptase is going to make one of the strands of DNA and then go back and make the second strand. And then from there, the DNA now, which is now carrying the genetic information for making new HIV viruses, is going to go into the nucleus. And the arrow's going the wrong way. In the prior edition, it was drawn the right way. I don't know why it's the wrong way. I checked in the book as well. So here your DNA now that is carrying the viral genome goes into the nucleus of the CD4 cell, and then integrates into a chromosome within there. And so that's going to require an enzyme called integrase, which was mentioned earlier. And from there, then we can now start transcribing the genes of the virus, of the HIV virus, right? So one of the things we're going to do is we're going to do a transcription there to make your mRNA. So your mRNA then comes out and binds with the ribosome, and you start making the proteins necessary for putting together new capsids and the envelope as well. So the proteins of the capsid, reverse transcriptase is also going to be made. All these things need to be packed within the capsid. And even the GP120 protein, glycoprotein spikes, are also going to go to the envelope. They go to the plasma membrane of the CD4 cell, and then the assembled capsid then goes to the membrane and then buds off. So now you're going to have a budding, and so this part of the plasma membrane will bud off, and so the virus takes a piece of the plasma membrane of the host cell with it. And so now here's your new HIV virus, and it can go and infect another CD4 cell. So that process of replication is it's a good example for showing a retrovirus, how a retrovirus works. That's exemplified by the HIV virus. Now, initially, macrophages would be infected, and then over time, mutations occur because it's an RNA virus. And one of the mutations occurs in the GP120 protein, so we get a slightly modified GP120, and that allows it to gain access to more kinds of cells of the immune system, and eventually that's going to be a T helper cell. And the problem here is that over time, once the T helper cells start becoming infected, for whatever reason, which is not well understood, the T helper cells, when they're releasing the new HIV viruses, the T helper cells end up dying in the process. And so, again, once you destroy those T cells, then you have these opportunistic infections that end up killing the individual. This is when they start showing AIDS, the acquired immunodeficiency syndrome. They're just not able to fight off other infections. So how do we treat HIV? The treatments today now involve a cocktail of several different drugs, and they refer to these types of treatments with this acronym HAART, which stands for Highly Active Antiretroviral Treatment. And they basically try to design drugs. There's many of these drugs that they've figured in using here. And basically you look at different points in the cycle of the replication cycle of HIV. And one is to block entry, right? So you might design drugs that block the receptors from going in, or even fusion inhibitors, so that if a virus does gain attachment to the right receptors, we're going to block the entry of the virus overall. So you can block entry in those two ways. And then if the virus does enter, you have some other drugs that would block or inhibit the action of this unique enzyme that's not found. It's not found in animal cells. This enzyme is unique to retroviruses, the reverse transcriptase. So you can get a drug that blocks the activity of that enzyme, then you can't reverse transcribe, so you block the cycle there. And then you might have other kinds of drugs that inhibit the integrase enzyme. So let's say you are able to reverse transcribe and you get your viral DNA now, and it goes into the nucleus, but then it cannot become incorporated into the host's chromosome, the host cell's chromosome. So you can block it there. Now, if you get past this point and the viral DNA integrates into the chromosome, then it gets transcribed. So the transcript, this is mRNA, goes out, binds to the ribosome, and you start to produce your polypeptide here. So there's your polypeptide there. That's going to become your protein. And the thing is, it's one long polypeptide. And that one long polypeptide has sections of it that become the different proteins. Some sections are going to become part of the capsid. Others are going to be the GP120 protein. Others may be the enzymes necessary for viral replication that the virus has to carry with it. And if you can't cleave up and break apart that long polypeptide, then you can never form the virus itself. So you would have drugs that block these proteases. A protease is a general term for an enzyme that breaks down a polypeptide. So that's the basics of HAART. Now, your book, especially the older edition, gives you all kinds of names for these different ones. It's not important that you understand or remember the names of these different drugs, but you should recognize that treating a disease like HIV requires drugs that are trying to prevent the replication cycle of HIV overall. Now, then we have latent infections, viral infections, and these are the ones that hide. They become proviruses. They become incorporated inside some tissue, for example, and they're latent there for a while, and they're not actively producing new virions to go infect other cells. An example of a latent one is the HSV virus, which stands for herpes simplex virus, and there's two types, type 1 and type 2. I think the type 1 is for the cold sores, and the HSV2 is the one that causes or affects the genitalia, causing lesions there or blisters. And those blisters are going to be filled with those, the fluids from those blisters are filled with these viruses, which can cause an infection when you come in contact with another individual. So these things are easily spread by contact, sexual contact, and so on. During periods of time when the virus comes out of latency. Now, during latency, the virus hides within the nervous tissue of your nervous system, and it's some trigger, perhaps stress or changes in hormone levels due to stress. So some physiological stimulus or stimuli are going to trigger the reactivation to come out of latency and then begin the replication cycle for these types of infections. So this specific example here comes from the one that causes cold sores and genital herpes is the name for the disease. Viruses can also lead to cancers, and one that has gained more attention in more recent times is one that can cause cervical cancer. Can also cause cancers in males as well. Cervix is part of the female reproductive system. In fact, 70% of cervical cancers can be traced back to the transmission of HPV, which stands for human papillomavirus, and it basically the papillomaviruses cause warts on your skin and so on, and this one is sexually transmitted, and if it gets into the reproductive tract and infects the mucosa in there, that can lead to cancers. And the reason it does that is because when the HPV becomes incorporated or integrated into the host's cells, into the individual cells of the reproductive tract, it can actually cause damage to cells that control the cell cycle. You might have covered this during the chapter on mitosis and the cell cycle in your Biology 1 class, but there are tumor suppressor genes, and there are proto-oncogenes. And the proto-oncogenes are genes that get the cell cycle to progress, to go through cell division. And tumor suppressors are the ones that put the brakes. So they're antagonistic of each other. And both are needed because sometimes you need cells to divide and sometimes you need them to stop dividing. it's when you lose control over that cancer occurs, right? And so that's the concern here. So we now have vaccines to prevent this, and this has been politically controversial in many circles, but that doesn't take away from the science. These vaccines can prevent infection, and that means they can prevent cancer later on in life. So it is recommended that the vaccine be given early on before individuals become sexually active. In this section, we're going to look at prions and viroids, and these are going to be infectious particles that are smaller than viruses. And so for your learning outcome, you should be able to explain how a prion replicates, and transmission doesn't necessarily follow what we normally see as involving heredity or genetic, the way DNA is involved in heredity and transmitting genetic information from one generation to the next. So for prions, prions is short for proteinaceous infectious particles. So here, these particles themselves are proteins, and they are responsible for diseases that are collectively called encephalopathies, or pathology or disease of the brain, your nervous system. So this is a spongiform because this causes the degeneration of nervous tissues, wherein the diseased brain looks like a sponge because so much of the tissue is wasted away. There's different types. There's bovine spongiform encephalopathy, which is what BSE stands for, also known as mad cow's disease. Then there's scrapie in sheep, and then Creutzfeldt-Jakob disease in humans, or CJD. Now here, the key here is that there's a protein itself that causes the disease. And so what happens here is that there's normal proteins found within cells of the nervous system, within nervous tissue. And these normal shaped proteins, which we're going to abbreviate as PRPC, these normally shaped end up coming in contact with a misfolded version of that protein. And that misfolded protein is going to be abbreviated as PRPSC. And so if this misshaped protein enters the tissue and gets into the cells where you normally have the normal protein, this particular protein causes the normal protein to misshape. And then you have this chain reaction. So over time, we start to build up a change from the normal prion protein to the misfolded prion protein. And then over time, that causes the degeneration of your nervous tissue. Now, some evidence for the fact that the protein is responsible for causing this, and not some particle that has a nucleic acid in it itself, like DNA or RNA, comes from working with mice. And so if you can engineer a mouse that produces that prion, we're going to call it the prion protein because of its association with prion disease, but the normal protein must have some function within the cell to begin with. They're just going to call it prion protein. But if you engineer a mouse to delete that protein, in other words, cause the gene to be defective so that it doesn't produce the protein at all, we're going to call those knockouts. And in the last, so these would be knockout mice, and they were knocked out for that prion. And for some reason, from the last book to this one, they changed it to the null mice. But this would be, we can also call this the deletion. So they deleted that gene from there. So this mice basically is able to live apparently without that protein. So it doesn't have the gene. So within the nervous system, these mice, this group of mice, it's a group, right? You have to have experimental group and control group in an experiment. These mice are living relatively normal, with normal activity. What they don't have, though, is that normal prion protein anymore, because we knocked it out. It's a knockout strain of mice. And then you have your normal control group that has that normal prion protein. We're going to call that the wild type. And so what they do is they get a mouse that's been infected with the misfolded prion protein, the PRPSC. And then they make a homogenate of its brain because this protein is found in the brain, in the nervous tissue. So they basically blend up the brain, chop it up real finely, and then they inject some of that homogenate into both groups of mice. And the mice that where the deletion occurred, where they don't have that protein, seem to do just fine. But the mice that are still have the normal prion gene end up dying with the neurological disorders that develop from prion disease. So this evidence here shows that it is a protein that's transmitting this this problem, and not some virus or a bacteria that has DNA in it. So that's a challenge to. Here we have a disease is caused by something that does not have DNA or RNA directly coding for some infectious agent. And then we have viroids, and viroids are tiny, naked circular pieces of RNA, probably not more than 400 nucleotides in size. And these are actually can cause disease in crops and plants, right? And so here, if this RNA, these viroids get into a plant through a break in the tissue of the plant, or an insect that goes and pierces the plant and sucks out the sap from there, it might take this viroid from one plant and transmit it to another. And once the viroid gets in there, it'll use the host plant cell's RNA polymerase to make copies of that RNA. And during this replication process, it's actually a much larger piece of RNA that's made, and then the RNA that's produced that becomes the viroid actually self-catalyzes breaks at very specific locations. So it's a much longer piece of RNA, and then RNA can serve like an enzyme. Remember, they have a ribozyme type function. So these RNA particles basically self-cleave or self-split themselves at the right parts, producing more viroids that can go and infect, cause more infection or more problems. During this time, when after the RNA is replicated with the RNA polymerase, and then the RNA cleaves itself, little pieces are produced during that time, and they go and they interfere with normal protein or normal gene expression in the plant, and that can cause problems with growth and development within the plants, and so that creates disease within the plants. These small little particles of RNA that are produced are called siRNAs. That refers to small interfering RNAs, and those small interfering RNAs have this right complementary combination that can go and attach to, say, a messenger RNA, and that would interfere with translation to make proteins necessary for the plant's metabolism. So there's sort of that mechanism for disease by these viroids.